And it is my pleasure to present Jonathan Scolnick from Singleron Biotechnologies, who is joining us from Singapore. So Jonathan is a general manager of Singleron in Singapore, and in the past he has 20 years of experience in research and development of genomic-based assays. He worked both academia and industry, among others in companies like Natera and NewGen Technologies. And most recently, Jonathan has been the co-founder and chief scientific officer of Proteona, also a company based in Singapore, where actually they were doing something very similar to what we do at Skylight, taking different single-cell approaches and combining them with tailored machine learning methods to develop clinical applications. And now they're doing this in the bigger context of Singleron, and Jonathan is here to tell us more about this. And I'm really excited to hear this, Jonathan. And thanks for joining.
Great. Yeah, thank you so much for inviting me, Peter. I really appreciate it. And I had some great talks in the first session, so I'm hoping I can live up to those. So I'm going to tell you a bit about Singleron Biotechnologies, and I'll start out by just telling you a bit about the company, because my guess is you haven't heard of us, even though it's quite a large and growing company. We focus on single-cell biology and really bringing single-cell biology into the clinic. So I'll walk through a little bit about some of our products that are driving the single-cell analysis into the clinic. And then at the end, I'll talk about a project that we had published in a poster at AACR this past year, which brings in then how we used single-cell biology and machine learning to do predictive analytics for predicting how our patients will respond to a particular drug. So if you think about single-cell biology, we really have to start with the tissue and take that through then to single-cell suspension, single-cell isolation, library generation, sequencing, and finally data analysis. At Singleron, we have products and services to handle this entire pipeline, although we use standard Illumina sequencing. As I mentioned, in addition to the products, we do perform end-to-end analysis services, both on our own platform as well as on the 10x Genomics platform. And that's really used for a bunch of different things like biomarker discovery, predictive models, as I mentioned, even biomarker validation. We have four major main sites around the world, Singapore being the most recent one, as Peter mentioned. The bulk of the company is based in Nanjing, where there are about 400 people. There's a site in Cologne, which has about 100 people and growing, and a smaller site in New Haven, Connecticut, which was actually where the technology for Singleron was developed. So we have active projects with 4 of the top 10 pharma companies across all major geographies. So we have samples shipped to us both in Singapore and Cologne from around the world. And then obviously the China office handles samples exclusively from China. And the applications, kind of as I alluded to, really range from diagnostics through translational research, actually reverse translational research in some cases, and more general disease research. And while we do work with pharma companies, the bulk of our work in terms of sample numbers really comes from working with academics and clinician scientists. We really love working on those types of projects because instead of just kind of the pharma company telling us, these are the things we think we want to do, we really get to dig in and work closely with the academic clinicians and really work on interesting projects and developing new tools and algorithms to help them in their research. So I showed this picture before where we have products in the space. So in thinking about clinical research in particular, one of the biggest problems we saw early on was that patients, or rather clinicians, can take a biopsy, and then how do you break that biopsy tissue down into a single cell suspension to be able to even measure the single cell gene expression. And so we spent time developing a product in that space, which we call the Python, which is a tissue dissociation machine. Then we have of course, our single cell gene expression analysis platform, which can be run manually or works on this instrument called the Matrix, the kits to go along with that to generate the sequencing libraries. And then we have a platform for bioinformatic analysis on the out on the end of that, which includes everything from sort of the most basic analysis all the way to using machine learning for predictive analytics. So as I mentioned, starting from sort of the left to right of that previous diagram, we spend a lot of time looking at tissue dissociation, and along with that comes tissue preservation. So some of the major challenges in the clinical space that we saw where do you dissociate the tissue? So if you're, say, an academic clinician scientist, maybe you take the biopsy yourself, even if you're a medical doctor, and then you bring it back to your lab and you can dissociate the cells in the lab, and maybe that works for you. But if you think about how a pharma company is going to run clinical trials, you think about that being across multiple sites, and you might want to actually do central processing of those samples because each individual site can't necessarily handle the cell dissociation or preservation, which might include cryopreservation of samples. You have to come up with a different solution to that. We also had problems, obviously, of working with limiting materials. So clinical biopsies can be very large, or they can be down to fine needle biopsies. And so how do you handle that sort of range of samples and being able to extract viable cells? And then there are a lot of tissues out there that people study, and a lot of them require different techniques for dissociation. A lot of optimization is needed. And so we spent time developing solutions for those dissociations. And we also work with people directly with their samples to try to optimize those techniques. So in terms of handling this question of sort of where or how do you handle your tissue if you need to send it to a central repository, or let's say often surgeries run late and the samples don't even get into the lab until 7 or 8 p.m. So we developed a preservative solution, which we call SC Live, for single cell live. And it can, basically you take your tissue, you put it in the solution, and that tissue is then stabilized at 4 degrees for up to 72 hours. It stays alive, and in a sense, it sort of freezes the gene expression. And so that allows you to transport tissue, say, within the continent. So within Europe, for example, you can easily transport tissue to a central site for processing. Or even, as I said, if samples come in, say, late on a Friday night, you don't actually have to have someone staying in the lab all night just to process the samples. You can actually store them in the refrigerator over the weekend. And then the Python instrument, which was pointed out on the previous slide, is the instrument that we use for the cell dissociation. So it's an automated instrument that is essentially a heated tissue grinder that comes along with an enzymatic solution that can be tailored specifically to different types of tissues. And this is just a little bit of data to show you that these systems work. So this is the SC Live reagent. As I mentioned, it comes in a little tube. You essentially dump your tissue into that tube, and it's stable. You can see just looking at the viability, fresh 24 hours, 72 hours later, the viability is still very high. And if you carry that then through to do a full single cell gene expression analysis, you can see that the samples are essentially overlapping each other. And you can actually look and see at the gene by gene basis that there's an extremely high correlation, even out to 72 hours of storage. The Python instrument itself is sort of easy to use, and importantly, it's very reproducible. So here is just looking at 3 examples of mouse kidney dissociations. So you can see the viability was similar across all of them. It's not shown here. The cell numbers extracted were also very similar. You carry through to the RNA sequencing again, you can see that the samples overlap. So there's no difference between, say, which channel you use on the machine, which I didn't mention actually runs 8 samples at a time. And you can see the cellular diversity is consistent across all of those samples. But now sort of the meat of what we look at is the single cell RNA sequencing. So we do that on what we call the scope chip. So this is a microfluidic device that is composed of tens of thousands or hundreds of thousands of microwells. So the cells are floated into the device. They fall into the wells just by gravity. And then we place beads on top of each of those wells. The beads themselves have then capture probes to capture the RNA. I'll get into that in the next slide, what the capture probes look like. We have two different chips. The standard chip is designed to collect about 10,000 cells per sample, and then a high density chip, which is designed to collect 30,000 cells. Now I mentioned those capture probes. We actually have a number of different types of capture probes, which gives us the ability to identify multiple types of RNA, not just standard mRNA profiling. But of course, there is mRNA profiling, which we can use then for single cell gene and single cell nucleus profiling. We have a special probe set for collecting TCR and BCR data. So basically, this is a probe that hybridizes to the constant region of a TCR or BCR sequence and lets us directly target those sequences. Similarly, in terms of targeting, we have a product line we call the Focuscope, where we have both pre-designed and we offer custom targeting panels. And so here we really use this for looking at specific regions of RNA, such as maybe targeting mutations or gene fusions. And we do that in the blood cancer panel and the lung cancer panel, again, where we can actually identify mutations or gene fusions from the RNA at the single cell level. We also have an off-the-shelf panel for looking at Epstein-Barr virus, so we can actually capture the viral RNAs, again, at the single cell level. So you can start to see precisely which cells are infected and how is that maybe impacting the gene expression. Because while we're collecting this information, of course, we're also collecting the gene expression information from the same cell. And then we have this product we call Dynascope. It's a little more of a research product, less clinical, but lets you really look at transcriptional dynamics. So identifying when a gene is actually being transcribed. So if we get into a little bit of data for how the scope chip works, the product for gene expression is called the Dynascope. And this is some comparison data to a commercially available platform that is commonly used. And so we can see here that in general, if you're looking at the gene distribution or the distribution of UMIs in cells, it's very comparable between the two platforms. And then focusing down here, given the same median reads per cell, we can see that our platform is very comparable, in this particular case, actually performing better than this platform X in terms of the median UMIs per cell, the total genes identified, and the median genes identified per cell. Because I mentioned the BCR and TCR kit, we actually, that targeted approach we use actually performs very well in comparison to non-targeted approaches or approaches that say capture the entire RNA population and then go on to try to target the BCR or TCR information. And so we can see that we can capture, in this case, looking at this, just the T cell receptor, 82.5% of T cells where we capture both TCR alpha and beta chains. And you can see why we're able to do that is really because with the direct targeting method, we just capture a lot more initial RNA molecules than can be captured in a total cDNA capture method. Okay, so enough about sort of how the technology works and what the products are. This to me is the much more interesting part, and it's a proof of concept study here where we were using our machine learning methods that we've been developing to try to predict patient response to this drug Daratumumab. So Daratumumab is a monoclonal antibody therapy used to treat multiple myeloma. And one of the reasons we had targeted this drug and sort of multiple myeloma in general is that in multiple myeloma, patients generally always relapse. So they go through multiple rounds of treatment, and each round of treatment can cost up to about 18,000 euros per patient per month. So these treatments are extremely expensive, and so it's quite important to try to understand which patients are going to respond to those treatments. So you're not, putting the patient through these treatments that they're not going to respond to, but also not requiring the healthcare system to pay these really high prices for treatments that aren't working. So here's the overall strategy we took. We had a training data set, which we actually took from publicly available data. So it was 10 patients who responded to this drug, Daratumumab, 7 patients who did not respond or were considered non-responders. And then we also followed that up. We had a test data set, which were patients that we had collected from here in Singapore. Anyone who's done work with machine learning knows that data cleaning is probably the most important aspect. And so we spent a lot of time trying to understand how to clean the data from single-cell RNA sequencing experiments. And here is just an example that shows sort of different methods we had used for data cleaning and how they impacted the model. And so you can see there's actually a very wide range of results depending on how we treat the data before it even gets to the model. From there, of course, we go on to make predictions. And then for anyone who was in Felipe's talk, he mentioned this thing that really rang true was that for their work over at Sophia Genetics, they had found very clearly that physicians essentially wouldn't believe their results if they couldn't understand why those results existed. So what is it within this data set that's informing the algorithm to predict whether a patient will respond or not respond? And so we also pay a lot of attention to that and make sure that all the algorithms we use are interpretable. And so we can actually understand what it is that the model is focusing on and sort of the weights of those different features. And in this particular case, the features are all genes. So again, we took this training data set from this publication in 2021, Cohen et al., where they had these 17 relapsed refractory subjects where they performed single-cell RNA sequencing on the tumor cells. And they performed that RNA sequencing prior to treatment with Daratumumab. And in multiple myeloma, you never just take one drug. It's always a cocktail of drugs. So in this case, it was Dara with this cocktail KRD. So we trained our model using a leave one out strategy. And then we predicted the response actually for each individual cell. And then what we've done here is just plot the distribution of those responses. And then because it's a very small data set, we've essentially used a heuristic to decide what a cutoff might look like. And so we chose a median prediction response score of 0.69 as our cutoff. So if you had a median prediction response score above that, we would predict that as a patient, you would respond to therapy. And if it was below that, we predicted as a patient, you would not respond to therapy. And so we correctly predicted 94% of the patients, badly missed this one. We're still trying to understand why that is. But I think there can be a number of reasons from technical to biological. And we don't have time really to dig in today to why that is. But as I mentioned, we also had a test set, which was taken from Singapore. I should say that published data set was taken from Israel. So there you had a mostly Caucasian population. Here now in the test set, we're looking at an actually exclusively Asian population, which is just a bit of added flavor, because if you look at experiments that just focus on genetics, for example, you always see that there are these large differences in how populations respond or to, say, a genetic test based upon where the original training data came from. But here we're showing actually across these two very different locations, our training data actually holds up. So we had slightly different labels here. Instead of responder and non-responder, we had a very good partial response, which is almost complete remission, and then compared to partial response, which is less than that. So you can easily identify tumor cells within the patient given a partial response. And so in this case, again, using the same cutoff, we were able to correctly predict all of the patient responses. And then just thought this was interesting, although likely not particularly meaningful, that we had this one patient that actually responded so poorly that they died in the middle of treatment, and they actually had the worst response prediction score. Now at this point, the model we figured could be on the one hand predicting who's going to respond to Dara treatment, but on the other hand, could just be predicting overall who is going to respond to any given therapy. So we looked at another data set where this was an independent data set where the patients did not receive Daratumumab. And then we could see that the model only predicted with a 66% accuracy of how the patients were going to respond. So that really suggested to us that the model is more predictive of the response of Daratumumab along with those other drugs that I mentioned. And it's not just sort of a general prognostic model. And then, as I mentioned earlier, of course, we can focus in on why the model is making these predictions. And this can be important, as Felipe mentioned, for convincing clinicians that the model is working. But in terms of reverse translational research, actually, this is really sort of great fun, because now we have this list of genes in this case where we can say, okay, patients who had gene number 1 upregulated tended to have a positive outcome and a positive prediction and a positive outcome. So now, that gene actually, in this case, none of these genes were known to be involved in multiple myeloma. So we can go back to the lab now and say, okay, if we have say a myeloma cell line, maybe we start to knock out some of these genes and see exactly what role they're playing in the disease. And so I'll wrap up here just to review that, basically we run the gamut from starting at the tissue biopsy itself, processing the tissue, getting it into single cell libraries, all the way through helping people with their bioinformatic analysis. We do that with a proprietary tissue preservation solution that allows you to take your tissue and save it or ship it to a central location for processing. Along with that, then we've developed this instrument, the Python, for actually doing the tissue dissociations in a consistent manner. Obviously, you need good cells, good healthy cells to start with your single cell experiments. We have a large menu of single cell applications. I mentioned a few of them. There actually are a few more that were a little less related to this talk that I didn't mention. And then we have developed this sort of deep data science expertise in trying to develop these predictive models for both a clinical setting, but as well as being able to do these reverse translation experiments so that people can go back into the lab and try to understand the disease better. So I'll stop there and take any questions that anyone has.
Thanks a lot, Jonathan. Very exciting talk and very interesting technology. And also the use case that you presented is very interesting. We have quite a few questions, so let's get started. I'll try also to follow the workflow that you presented. And I think it's really important to understand the importance of the quality of the sample, starting from collecting the sample and then all the way up to generating the data. And actually, when you work with the data, also there you need to pay attention to what you're doing to actually generate the insights. So it starts from the beginning, from the patient. And on this note, you mentioned that you presented your sample preservation reagent. So there was one question, is it working both on solid and liquid samples?
Yeah, so it actually exclusively works on solid samples. So for liquid tumors, for example, or PBMCs, we would still suggest cryopreservation. And this solution is really meant for solid tissue preservation.
Okay, good. So we clarified this. Yeah, then there was moving to the questions on the data cleaning. So you made the point that it's really important kind of to try different methods. So can you elaborate on how did you approach the feature selection and the regularization when you for your predictive single-cell RNA-seq models?
Yeah, so initially we just kind of threw the kitchen sink at it. It was nothing special in that regard. The general machine learning algorithms are all out there and available. We're not inventing anything new, and it's really the focus on the data cleaning and sort of understanding the biology behind the disease. I would say was also very helpful because there were specific features where, because we understood the disease, we knew actually that we didn't want to include those features. And actually taking those features out improved the models tremendously. So it's not just having the data scientists who can really drive that aspect of it, but also keeping the biologist involved in every step so that they're there to pay attention and say, oh, wait a second, actually, you should get rid of that feature, or make sure we bring in this other clinical feature that we maybe hadn't thought of.
Yeah, that touches upon the multidisciplinary approach also that Christopher is pursuing, where again it's not only on the computational side where you need to work and design these experiments, but you need to understand also the biology. So yeah, thanks for this answer. Also related to this, so when you did these experiments with trying out different methods and data cleaning, and you've seen actually different outcome on the models, what were the major factors that had an impact on the interpretation of the data that you've observed?
So the one that I thought was the craziest was actually just the method used for mapping the sequencing data back to the genome. I was really surprised that had a very large impact on our model. That's one of those things that at least on the RNA sequencing side, I know in genome sequencing, it's extremely important to understand, exactly which mapping method you're using. But I kind of thought on the RNA sequencing side, it didn't matter so much. And I was definitely wrong. So
okay, that's a really interesting observation. So there is another question, which actually also we at Skylight get quite often when we talk to other people. It's from Lorenzo, and he's asking, can you comment on the issue of training machine learning algorithms with 17 samples and thousands of features? How do you avoid overfit?
Yeah, good question. So we do start by doing some feature reduction. And in this case, for example, because the training data was only performed on the tumor cells, that actually reduced the potential feature space considerably compared to, say, a sample of like PBMCs where we would have had a lot of different types of cells. So you would have just really been multiplying the number of features. But we can get rid of a lot of features right off the bat, things that have very low expression across all the cells, or things that have just completely even expression across all of the cells. So sort of little tricks like that.
Yeah, that's on the reduction of the number of features, but in terms of sample size, so working with sample sizes of 15 to 20, isn't this limiting, or how is this working, and how can you validate this later on another cohort?
Yeah, it's definitely limiting. Of course, we would always love to have bigger data sets. And, unfortunately in this space, bigger data sets costs a lot more money. That's actually, one of the reasons you mentioned was previously with Proteona. And when we were acquired by Singleron, that was one of the reasons that we wanted to work with them, was because all of a sudden we had a much better handle on the cost structure. So we could reduce our costs considerably more by also making the tools to collect the single cell information. I would say that going into the experiments, we obviously were very concerned that 17 samples was going to be too small. It was, all we were going to see was overfitting, and that was going to be the end of it. But what we've seen now is actually, even with a small data set, I showed a little bit of a test data set there, which again was a completely independent cohort, taken at different time, different place, with different methods. Actually, the gene expression itself was measured using a completely different method. And we could see that actually we could make pretty good predictions. And we've continued to collect samples from DARA-treated patients, and the model has continued to hold up. So I would have said in the beginning, for sure, I totally agree, 17 samples seemed like it was too small. But it's kind of like the little engine that could. It sort of keeps going. Okay. Of course, I would say, a major caveat going into if we were going to take this into a clinical setting, we would do a much, much larger study. Certainly wouldn't rely on such a small data set for that. But for research purposes, I think what we've seen is you can get a lot of very useful information out of a small-ish data set.
Yeah, and this is also our observation, that using single cell data, which is really high dimensional, and also if you use single cell RNA-seq, which is unbiased, so you measure all the genes that are in there, even on small sample sizes, you have really big discovery power. So, and that is specific to single cell RNA-seq data. There is actually a question from Corinne. So how do you correct for potential lab-to-lab heterogeneity when using public datasets for training your model and proprietary data for validation? So the lab-to-lab heterogeneity, and you mentioned this a little bit. So one way to generate more data is also generate at multiple sites, but then how do you correct for the heterogeneity there?
Yeah, so there it depends a little bit on the question we're asking. So we have developed specific batch correction tools that can address exactly that problem. But what we found is actually these models seem to do the correcting themselves. And I know that's not a very satisfying answer, but that's just kind of what we're seeing, is actually the variation from site to site is essentially taken into account when the model is weighting the different features.
Right. And a question from Sarah. So did you look into which cells or cell types were correctly predicted versus incorrectly predicted within each patient? And were there any interesting patterns?
Yes. So these cells were all tumor cells. And so we haven't really been able to break it down further than that. With the training the training data set, again, because it was publicly available, that's kind of what we had, and it was all one cell type.
All right. So I don't see any more questions from the audience, and also I asked my questions that I had. So I think with this we can wrap up this session. So I hand it over to you, Eleni, and again thanks Jonathan for this really interesting presentation and the Q&A session.